Process of transcription. How RNA that is formed, especially the mRNA from the DNA. Only one strand that is involved in this transcription. One strand of DNA that is involved, the 3 to 5 minute direction. But RNA always synthesized in 5 to prime end direction only. Either new DNA or new RNA that synthesized in 5 to 3 end direction only. The procedure or the process of transcription requires, what it requires? Enzymes. Enzymes. Here also helicases required. The main enzyme DNA dependent RNA polymerase, topoisomerases also required. Topoisomerase also required. Next, nucleotides, ribonucleotides, ribonucleotides. It is also energy consuming process. It is also energy consuming process. Ribonucleoside triphosphates, that is the ATP, GTP, CTP, UTP. Adenosine triphosphate, guanosine triphosphate, cytidine triphosphate and uridine triphosphate. Adenosine triphosphate, that means the ribonucleoside triphosphate required. Factors. Sigma factor and rho factor. Sigma factor and rho factor, these two are required. So, different requirements for this process of transcription and the procedure. So, in the longer DNA, only some segment or only some part of longer DNA that involved in transcription. In this region, the two strands are unwound by the enzyme activity helicases. The two strands unwinds 3, 5. So, only one strand that acts. RNA primer. That is essential for the initiation of transcription. RNA primer is essential for the initiation of transcription. RNA polymerase, that is the DNA dependent RNA polymerase attached to the RNA primer. For this initiation, factor is required, that is sigma factor. Sigma factor is required. RNA primer, that is essential for the attachment of RNA polymerase. Anchorage for RNA polymerase, RNA primer is required. Now, according to code sequence present on the template strand, it is the template strand. nucleotides are attached to one another, that is the polymerization of the nucleotides that occur. Polymerization of nucleotides that are added. So, chain of RNA that is formed. Chain of RNA that is formed in 5 to 3 end direction only. 5 to 3 end direction only. When it completes or when it reaches to the terminator point or termination point recognized by rho factor. Initiation site recognized by sigma factor, termination site recognized by rho factor. The RNA that is first formed called primary transcription. That is called hnRNA, heterogeneous nuclear RNA. Why it is called heterogeneous nuclear RNA? Because it consists of two types of regions, introns and exons. Introns and exons. hnRNA consists exons because the template strand consists of split genes. Split genes, these are the regions where unrequired sequence present intervened with required sequences. That means some nitrogen based sequence are not required, called introns, and some nitrogen based sequences are required, they are involved in the coding, called exons. Split genes are those genes which consist required nitrogen based sequences interspread with unrequired nitrogen based sequences. In this mRNA that is or the RNA that is formed from this DNA strand or the template strand consider these two regions, unrequired sequences called introns and required sequences called exons. So, RNA with these two introns and exons called hnRNA. It must be processed for the formation of the functional mRNA. So, hnRNA next entered into the processing. The name of processing, that is the removal of the introns, that is called splicing. Removal of introns from mRNA called splicing. So, now the hnRNA, it is hnRNA. Is the 5' and 3' end. In consists intron, introns, interspread with exon, exons, unrequired sequences, introns, required sequences, exons. Removal of this introns, that is called splicing. For splicing, splicing complex involved. A splicing complex consists small nucleolar RNA plus proteins, collectively called as small ribonucleolar proteins. Simply say small SNRUPS. SNRPs. Small nuclear ribonucleoproteins. SNRPs, simply say, generally say SNRPs. Now, these small ribonuclear proteins, they are attached on either side of this introns. Called spliceosome. So, a spliceosome contains a small nucleolar RNA and protein. Simply say, small ribonucleolar proteins, SNRPs, attach on either side of introns and remove that introns. So, finally, process structure is formed. So, without introns, and these two exons are joined by ligase. So, in before this splicing, at the 5' end, capping that occurs. At the 5' end, capping. Unusual nitrogen base that attached to the 5' end, that is methyl guanosine triphosphate. Methyl guanosine triphosphate attached to the 5' end. Many adenines, that is called polyadenylation. That cause tailing. So, before processing, the first step is the capping. Unusual nitrogen base that attached to the 5' end of the hnRNA, that is called capping, and that one is called methyl guanosine triphosphate. And then many adenines that are attached to the 3' end without template, without independent template, that is called polyadenylation. That process is called tailing. After that, splicing is occur. So, the sequence of processing, sequence of steps in processing, sequence of steps in processing. First capping, next tailing, next splicing. Capping, methyl guanosine triphosphate. Tailing, many adenines. Splicing, removal of introns. Sequence of processing or sequence of the various steps during the conversion of hnRNA into the functional mRNA. Capping, tailing, splicing. It is the process of the formation of RNA or mRNA, or the process of transcription. Now, it is the mRNA. After processing, hnRNA that becomes the functional mRNA.